Good morning. Good morning. Did you all sleep well? Yes. Last night I was looking out the window and I saw many debaters. I saw many debaters out over there. Were you debating? Yeah. How many debated? How many won their debate? How many won their debate? One. Do you win a debate? You lost? Oh, it was a draw. A draw. So today we're going to be talking about chemical transmission. But before we get into that part of the lecture, I wanted to talk to you about something many of you have asked. And that is, how do you learn? Is this something of interest? Yes. Good. So, to tell you how we start to learn something, I have to make a few definitions. First, there's something called short-term memory. And something called long-term memory. Short-term memory may not last very long. For example, you may have forgotten what you ate 2 weeks ago. But most of you remember the day that you were born, or the year that you were born. Okay? So that's the difference between short-term memory and long-term memory. Also, there is memories that you are aware of. We call them conscious memories. And memories that you are unaware of. Let me give you some examples. Yes, I didn't want to use those terms, but okay. So let me give you some examples of conscious memories and unconscious memories. Conscious memories are memories you can talk about. For dates, for people's names. For things that you learn in classroom. You're aware of these kinds of memories and you can talk about them. So, what year were you born?
kilometers. Did you get 1985?
Okay, conscious memory.
What is his name? Uh-oh. See?
Conscious memory. Where was the Dalai Lama born? Okay, conscious memory. What is the capital of Japan? Conscious memories. You understand this. What you read last night, read 2 weeks ago, conscious memories. Okay? Now, unconscious memories. Do you have a bicycle? No. Who has a bicycle? No one has a bicycle. Do you have a bicycle? No. How many of you know how to ride a bike? Okay. Okay, everyone's hands go good. Okay, so now I talked about this yesterday or the day before. You know how to ride a bike? Yes. Do you remember when you learned? You do? Okay. Conscious memory. Do you remember the color of the bike?
Black.
Black. Conscious memory, unconscious memory. Now, he had to learn how to ride his bike. Who taught you?
His classmate.
His classmate, okay? Conscious memory. His classmate held the bike, he got on, okay? And the classmate pushed him. Okay? And he fell over. Conscious memories still. But after he practiced for 2 days, 3 days, he learned how to move his legs. More important, he learned his balance. Okay? So, was that learning conscious or unconscious? He remembers the day that he was riding the bike. But he doesn't know how his legs remember or how he remembers the balance. Many things that you learn, you're not aware of learning. Those are unconscious memories. And they get, if they're, for example, learning of motor skills, they get stored in a part of the brain the basal ganglia.
이거 가서 그렇으나 이제 베이सल ganglia 레션이 봐요. 그래서 레벨이
So, you remember you were telling me that one of the functions of the hippocampus was to help you make memories. But it's only for memories that you are conscious of. It's not for memories like learning to ride a bike, unconscious. How do we know that? Give me an experiment.
So we can see whether there are like electrical activities in the hippocampus during recollection of implicit memories.
Okay, okay. Very good. The way that scientists learned this was by a medical accident. There was a young man, he was a boy, in the state of Connecticut where I live. When he was a little boy, he was on his bicycle and he fell off and he hit his head. Okay? Some people who have a bad head injury develop epilepsy.
Epilepsy is the
seizure disorder.
They usually shake very strong, fall to the ground, and lose consciousness. That was due to the head injury. Because when he fell, okay, his brain hit his skull. Brain, skull. And the other side. It damaged his hippocampus. Initially, they treated him with medications that to block the seizures. And this worked for a while, but after a while his seizures returned and got more, and he had many, many more seizures. He went to a doctor who said, when you hit your head, you damaged your hippocampus. And that, we know, leads to seizures. I want to try a new surgery.
I want to try.
I want to remove your hippocampus because it's scarred. They were so severe that he and his family agreed. So they went into his brain and took out hippocampus on the left side, hippocampus on the right side. And then what happened?
So he had implicit memory but not explicit memory. Newly formed, he could not newly form explicit memories.
His seizures got much better, but he could not form new memories for facts and events. When they tested his ability to learn motor skills, they gave him something he had to do with a pencil and a piece of paper. And they were wondering whether over 5 days would he get better at doing this exercise. Did he get better? Motor. He did. But what's really fascinating is each day when he came in to his test, he did not recognize the person. He did not recognize the piece of paper, what the instructions were. So each day he was able to read, but he did not remember what the instructions were. Because those are conscious memories.
And he couldn't form.
So he was able to learn the motor skills, but could not form new memories. Okay? So this is a clear separation of conscious memories and unconscious, in this case, motor skills. Now I'm going to tell you, I told you earlier about short-term memories and how you turn them into long-term memories. You will learn much more about this next year. But I want you to get a sense of what has to happen for you to start to learn. So as we've said many, many times, all of your actions, all of your thoughts requires synaptic contact. Really neuronal contact. So as you learn, the contact gets stronger.
떼어주고
So let's say this was a particular contact that was going to help you ride a bicycle.
다음에 먼저 광고를 따야라 먼저 광고를 따야라 다음에 이거를 고양이다 와서 저분이 바로 있다 대충 이거 있나?
Initially, when you start to learn the bicycle,
광고에 광고를 따야 장애 보트도 상관
you only have one synaptic contact.
다음에 와서 니 이거를 다음에 와서 저분이 다음에 제줄 찍어도 그랬어.
As you start learning, you start to make additional contacts.
다음에 광고 따야 되고 무슨 야를 딱 장대 상관 그니 바로 딱 뒤줄되는 말을 그랬어.
So another axon comes and make contact with this neuron. So now you have two. As he practiced, he got three and four. So when this neuron fired an action potential, this neuron got a big input, very strong input from now 4 different axon tips. So this is one way that you can strengthen the contacts, is to add additional synapses. There's a second way that I will mention now, and you'll learn more about it. When you want to strengthen this synaptic contact, what has to happen? When you want this action potential to pass to this neuron, what must happen? You must release neurotransmitter. And that's what we will talk about today. And as you strengthen this contact, when an action potential, as you learn, when an action potential arrives, one way that you can strengthen the contact is to release more neurotransmitter. Or put more receptors here. Okay, so I've mentioned 3 ways that you can strengthen synaptic contact. One, more neurotransmitters are released. 2, more receptors over here, postsynaptic. 3, more axons from here. Okay, these 3 ways. Okay, that's enough for now. But the important point is, as you learn, you must strengthen neuronal contact. So this is the outline of today's lecture. To understand how neurons communicate with other neurons or with muscle, and to understand the role of neurotransmitters and receptors in this process.
Sorry, could you repeat it?
And to understand the roles of neurotransmitters and receptors.
receptors in
How you, in the process of neurotransmission.
다이네 원자 조금 바르기다 진동들라 네 원자 조금이다 원자 증발이 이렇게 증발이 가는 차례인데 딱 하고야 돼. 나들이 이제 녹지들이다.
So is this say chemical neurotransmission?
No, it says the goal of this
Oh, this is what I just said. Okay, fine. Which? Okay, fine. So neurotransmission is how neurons can communicate with each other.
저가 원자기 다 진동 제한제 원자기 진동 제한이었으나 원자 저번 지지 거를 다 변지고 고다체단들이
Is required, of course, for all your actions and your thoughts.
저자다에 가서 자와다 다에 삼고 건드려지 바이나 니버도 보여지 차도레스
For the beating of your heart and your breathing.
다에 님 나와다 님 파르다 다에 신경 우다야라야 보레스
Okay, for all of those, neuronal communication is required.
저자다에 원자기 이름이 상말로라 이제 기타 진동이 니버도 상고해지 차도레스
And many toxins disrupt this process.
다에 두 망고지 다에 이름이 가서다 가고레스
Many drugs that we've developed also improve sometimes the communication.
다이네 사람들 다이네 서베다 맹망고치 다 왕자기 맹망고치기 다 디네 야라타고 그랬어 이 이름 야라타고 그랬어
And if it's defective, it can lead to a number of different diseases.
다 이름이 그때 뛰어 차와이나 다 나사 그랬어
Okay, so you've seen this kind of cartoon before.
네 빠르게 니마 토니 와스
These are called
내가 가서 그래 나게 난 날린 사람이 나게 사람이 dendrites axon dendrite terminal axon terminal
axon terminal.
나게 사람이 준비해봐
Okay? So, what is happening here this time that we want to emphasize is synaptic contacts
다이네 드링감서 다 surgeon 쳐봐야 돼 가르스나 드링이 롭쳐가 쳐봐야 돼 근데 나게 사람이 날린 사람이 라 가르스나 원자 저번 셔봐 이따 뒤섬기 다 뒤줄지라
synapses are occurring throughout the dendrites.
다이네 뒤섬들 가르스나 그 날린 사람 상관없고 켜대우라
Giving electrical information, electrical signals. These electrical signals travel down to the cell body. If there are many of them, they get sufficiently big to start an action potential to go down this. Okay, so what we say is that these electrical signals are integrated or added together. And if they are big enough, when they arrive at this point, if they're big enough, an action potential is started. This part of the neuron has a specific name. We call it the axon hillock. So yesterday, David talked a lot about the action potential. And he was talking about the coming in of an impulse, the arrival of an impulse,
그 로기 같은 데에 그 조당 매니지 있는 라브레스. 파르덕 된데 간지 그 준비를 라브레스.
So the David's action potential arrives here,
저상가에 레지 톱스데와 렙체
gets integrated here, and
action potential.
I'm sorry, there's an arrival of an electrical signal. Yeah.
저상가 로기 같은 데에 달린 사진의 조르체.
And right at the axon hillock is where the new action potential is generated. Okay? And so then as this new action potential travels, what are three proteins that are necessary in the cell membrane? What was the first one?
Potassium channel, sodium channel, potassium channel, pump.
Where's my pump?
Yes. Pump.
Who was the pump yesterday?
Which transmitter 가자.
Oh, there's my pump.
Which was?
Okay, that establishes the resting membrane potential. When this, there's enough of a signal arriving here at the axon hillock, it opens up which channel?
Sodium rushes out.
Rushes out.
I mean, sorry, sodium rushes in. And with a brief delay, what happens? And what happens? And that rapidly goes down this axon. It comes to a new neuron. And makes synaptic contact with the neuron on their dendrites. Here's another picture. Here we have the neuron, here we have an action potential traveling down the axon. In this case, it's making 3 contacts. And something new is happening here, chemical neurotransmission. To understand what is happening here, we have to fly into this synapse. Okay? So, let me identify some of the players here. What is this? Okay, it's an axon, and an action potential is arriving. What are these little guys?
Mitochondria.
What does mitochondria make?
Energy.
In the form of?
ATP.
Right? Were you eating ATP? Were you eating ATP? Okay, to function the pump. These mitochondria were responsible for making the ATP?
저단데 느껴 원타 되게 느껴 원타 매바이나 근데 ATP에서 두 번레스.
What are these things, these purple things?
네 무멘 되게 가래. 무멘. 너 무멘이다 무멘.
When you build a house,
말에 무멘 되게 가래.
what do you put in the walls?
저단 기록적 컴바이 접대 달라스 네 집바가 가리 잡고레스. 자, some wire, some metal wires.
Or bricks to hold the shape of the building. And in this case, these are structural elements.
저단도 컴바이 접대 정말 컴바 거라기 속다데 심리적으로 속다데네 그 심통리적으로 어디 저단에 단다기 무멘 동무멘 단다비 리무들이 주도 요건으로 어디가스나네 원자 조금 거라기 있다는 쿠바 좀 있다는 제들이 그래서 그래서 시험 봐
And there are other ones that are not in this picture, but are little railway roads, tracks. How does mitochondria get here? Where is the mitochondria made, first of all? Where is it made?
Some say nucleus, some say outside nucleus.
Yes, okay. Very good. It's outside the nucleus. So what? The various there are many, 100 proteins that go into the making of one of these mitochondria. This is a brief review of what you learned a couple of years ago. You remember the protein is encoded in the gene for that protein. There's a gene for protein A that gets transcribed into what? DNA to RNA. Okay, that happens in the nucleus. That RNA, we call it a messenger RNA, gets brought to the outside of the nucleus. Another particular molecule hops onto the RNA and translate it. Do you remember what that assembly is called that hops on? Ribosomes, okay? And it produces the protein. And many of these proteins are brought together and they then form a mitochondria. But my question was, how does it get from the cell body all the way down to the axon tip? How does it go there? Okay, I need a mitochondria. A mitochondria, come here.
Okay, stand here.
This is a mitochondria. Okay?
드링 느낌 원차다.
What will they be making?
분석해서 그래? ATP.
How you say ATP? Dupa?
ATP?
How do you say it?
ATP? ATP we don't translate, just ATP.
So, come over here. This is the we're right here outside the nucleus. Okay, the nucleus is over there. The mitochondria has just been assembled right outside. And they have to go all the way down there to the axon tip. So I mentioned earlier that there is a railroad track. Okay, right here. And there are proteins that can walk down this railroad track. They look like...
I think we have a good animation, right?
Do you have a good animation of this?
Research on YouTube, there's a good animation.
Okay, well that'll take some time. So, can you do that?
The Harvard animation of the cell.
Okay, we'll look for it and we'll bring it later. I'm a good animation. Okay, so I'm the protein that transports mitochondria. Okay, so now you know how mitochondria get transported down this railroad track. I also can, okay, so now I put you, now you're in the axon tip, you're this guy here, and you're busy making ATP. But everything that is here, every protein that is here, has to be transported. Okay? Is this clear? So what is this here?
Vesicle.
Okay.
Neurotransmitter vesicle.
How does a vesicle get down to the axis? The same way. And everything you see that is made up there has to be transported down this railroad track. Okay, so here we have a synapse. What is this region called?
Presynaptic terminal.
And this is the?
Postsynaptic terminal.
Yes. And what is this space called?
Synaptic cleft.
Correct. So now an action potential has arrived. So what happens to the potential here, the potential in this region?
Becomes more positive.
Exactly, it becomes more positive. A big surge of positive ions arrive. It has an effect on certain proteins that are also called ion channels. And those ion channels open. And more ions flow through. One of the very important ions that flows through is calcium.
다음에 드릴도 다음에 기들 디디 나오는 개치 휴직 내다 칼슘 사이 기들 레스 칼슘 스테로는 다 루고다는 드릴 주사기 제대로.
Okay? And what calcium does, it helps to bring the vesicle to the membrane.
근데 칼슘기는 랭게 가르스나 네 가모디 있고 기목이 있어도
Calcium is an ion. It doesn't do this by itself. It activates proteins, other proteins.
저는 칼슘 코랑 가르스나 네 기들레스 기들레 뭐 간식라고 딱 기름이 레가도 코랑이 치고 말에 네 주제 전반치가 딱 그마다 뭐 그긴 치고 그랬어.
And what do you see happening now?
어디서 가르쳐 버려?
The vesicle is fused.
Fused into the membrane.
And releasing the neurotransmitters.
Okay.
This is just saying what I just said, that if we look at these, before the action potential, this ion channel is closed. With the arrival of the action potential, this channel opens. And in flows an ion, in this case calcium. Okay, so I'm going to now show you a movie because I want you to fly right into this synaptic cleft. Movie time. Okay, we can all fit. Just turn sideways. Okay. Zoom. Okay, we're going flying up in here. Okay, so now we've come up into the synaptic cleft. Right? What is this structure?
Postsynaptic terminal.
This is the postsynaptic. This is presynaptic terminal. And it's releasing neurotransmitters. Some of these, there are many proteins that are in the membrane at the postsynaptic terminal. And some of them, the red ones, are receptors. So let's say that we want to, just a little review now, we want to strengthen this synaptic contact. Three ways we could do it. One. Okay. That would be out. We're too close. We couldn't see those coming. But that's right. The second way.
More neurotransmitters.
Where do they get released? So as you learn, more neurotransmitter might be released. What's another way?
More receptors.
More of these red molecules. that get inserted into the membrane.
That
Get inserted, put into the membrane.
저단디 네 키모다 내그로.
Okay? There's actually yet another way that you can strengthen this contact.
저단다 딜리로 뒤줄 때때로 따이네 도금 심바지레스.
You could make these receptors more sensitive to the neurotransmitter.
따이네 넬렘바디 완전 침바디 서로 낌바 소야들레스 서로 낌바 소야들레스.
So there are actually several ways that you can strengthen the synaptic contacts.
저단다 이제 딜리로 뒤줄게다 뒤줄 때때로 따이네 도금으로 망보레스. 가시지레스.
Okay, so here again are some neurotransmitters.
따뜻 완전 침바디 이들레스.
I've shown you some of these before.
정말 가시지 기능서도 때 마스.
Glutamate.
Glutamate receptor.
Does it excite or inhibit?
Glutamate 나르단치고라 감동치고레스.
GABA, excite or inhibit?
야바. 나르단치고라.
Acetylcholine, what does it do?
가르치고레스.
Does anyone remember acetylcholine?
아세틸콜린 재미도레스.
It is released at the muscle.
어디에 있는 레서드 때문에 셔름
It's the neurotransmitter that makes your muscle contract.
어디에 있는 그 셔름데 구미야 라카스 아세틸콜린데 있는 로그레스.
We'll be talking a lot more about acetylcholine in a little while.
아세틸콜린 디코르가 에네 테스젤라 게시 요시기스.
But I'm just mentioning some other ones that are very important. Dopamine,
친구 딱 언제 좀 하셔거든요 그지레스.
norepinephrine,
Dopamine, Norepinephrine,
and serotonin. Okay, so for example, why is serotonin so important?
세로토닌 계시면 잘 견적을 했으나
One example of what it helps with is when you're falling asleep.
또 경험수 니코드 참바 네 따디 니고 세로토닌 계시면 잘 했으나
It helps to quiet the brain.
그다음에 그래서는 레바데 쿠스 무서야 내가 싶어레스.
Norepinephrine
다음에 지금 Norepinephrine 에리아 먼저 좀 봐야겠어.
helps to activate you for action.
다음에 Norepinephrine 그래서는 기능하는 수부대 다시 그룹부터 볼까.
Look, there's a snake. Okay. So you then get ready to move, to run. Or you're excited for whatever reason. So, dopamine is very important for movements. It's produced in a part of the brain, one part of the brain called the substantia nigra. What does that mean? It means black substance. Substantia nigra. It produces dopamine, and the neurons that come from the substantia nigra go up to the basal ganglia. So it goes from the substantia nigra up to the basal ganglia that regulates your movements. If you destroy the substantia nigra, no, not brainstem. We're talking about the basal ganglia. So dopamine from the substantia nigra goes up into the basal ganglia and is required for motor movements.
저사다 도파민 가르스나 ene leşit zadı lepçe ene vücuda ene tanzı çeviriz. Vücuda keçimi çeviriz.
If you destroy the substantia nigra, what happens to the dopamine neurons?
다 ene zel 나고서 ene 그 ene 견쇼스나 같이 그래서 그 도파민과 같이. 소통하res. No dopamine.
Exactly. Okay? And in fact, if you destroy it, the black substance, substantia nigra, looks white.
White. 다 ene 제 나고데 그 torso tor와 있느라 그제도 가고 차고 그래서.
Many different movement disorders occur in the basal ganglia.
저사다 ene vücudun rivayette çünkü mamboçi da ene da leşit dilay çumores.
One of the most common, and you probably have seen people who have this, is Parkinson's disease.
다들 다 충격이나 리치 가서 버렸다 ene 먼저 mamboçi desay다 mamboçi 곧 desay다 충격이 그래서나 Parkinson 기타 충격을 했다.
So I'm now going to show you someone who has Parkinson's disease.
Parkinson di 충격이 여기다 미기 vücut tantan çevresi var. This çevresi.
Okay, severe Parkinson's disease. Has anyone seen someone with Parkinson's disease?
Parkinson di 충격들이 나데기 일단 미들이 통용해서.
Yeah? You've seen? Yeah. And what happens is that these neurons, dopaminergic neurons that make dopamine, is destroyed.
다음에 들리려 있다 도파민 기타 완사 저건 되게 듣고 있긴 쇼스나 같이 그래서. The movement will be impaired. Right, the movement will be impaired.
Yes, and they cannot do a smooth movement.
저산다 구조되는 직가소를 구조대 구치마주신 제마도 같다 구조 직가소보다 충분하다.
Sometimes they have trouble starting their movements.
다나신 구조 고주야가 kokang이 되겠어.
And they also have what we call pill move. They shake their hands like this. A pill rolling.
다나신 코단데 누구 주스 주스
There are other diseases, since we're on this topic,
많이 기타 충전 셔버들 그래서.
from the basal ganglia.
네, 레슈 자도 안 들어가야겠다. 네, 충전 셔버들 그래서.
So, for example, have you ever heard of Tourette syndrome? Tourette, okay,
Tourette 충전 해야 될 걸로 보면서.
is a tic disorder.
틱.
Tic. Okay, I'm going to show you an example of a tic.
다음에 틱스야겠다. 네, 근데 충전 주의사다.
It can be very mild
네, 데스카소 지금 참고됩니다.
or severe.
앞에 잡친구.
If you have the tic and vocalization, yeah, okay. If you have a tic disorder, movement of the muscle, if the diaphragm has a tic,
저한테 중증 조사에 들어가면서 나는 저기 diagrams 때문에 우당 사이 로이드 원나 지금 원고디들 그 중증 조사에 대해서 그랬어.
most of you have seen people who have a tic, right? How many have seen people with tics?
어디를 나서 나킨님이 통용했어.
Has anyone seen someone who has the motor movement and the vocalization?
네, 그래서 구조기 샤름기 구조들이다 다 하시면서 기체기 구조들이다 중증 나는 쇼딩인데 통화했어.
Did you see someone with both?
여기까지 나한테 기체식을 잡았던데.
So initially, if you don't know about movement disorders, when you see someone who walks down the street with a tic disorder, what do you think?
저단다에 기능서 단다에 구juk 있다네 주경 되는 거라네 장 만용 켄지 바이나다네 람가가 되는 다시 시간에 남겨 킨서 되는 통통스메 기능서 같이 그랬어. 영어에서 그랬어. You will think he's crazy.
Exactly. You think they're crazy.
영어에서 그랬어.
Now you know a little bit more that they're not crazy.
저단 기능서 단다 장차로와 대물로 장차로 선사에 꼭 부족 영어 많이 받고
They have a movement disorder,
부족 구족이 주경들이 그랬어.
and the part of the brain that is not functioning well is the basal ganglia.
저단다에 그 레벨이 제시 그 교수사대 그랬으나 그 내십사도 잘 그랬어.
We now have some medications we can give. So before medication, and then I take the medication. Okay, so the medications improve the tic disorders. Okay, a little diversion.
This cosmos.
Okay, so this is summarizing what we've already talked about. So here we have 3 axons arriving. The action potential is arriving at the axon tip. Depolarization occurs here. In the dendrite. Travels, and it may decrease a little bit because it's traveling. But if we have multiple depolarizations coming in from different dendrites, or a whole number of them arriving here, so that this is larger, then when it arrives here, what is that region called? The axon hillock. What is begun? What is initiated? And here you see again something that David mentioned. What is this here?
It's a myelin sheath.
Exactly, a myelin sheath. And what's important to know or let me ask you a question. You know that you have a massive depolarization here. Sodium comes in, potassium rushes out, and then it moves very quickly down here, the speed of light, and then you have another depolarization here. So one last comment, and then David wants to add something. So here, where do you need to have the sodium potassium ATPase, the pump, and the sodium channel, ion channel, and the potassium ion channel? Do you need them here?
Did you ask the pump or channel?
All the three of Will they be here? They're not there. No. They're here. They're here. They're here. They're here. Like the exercise we did yesterday. Do you remember the exercise? Okay, David.
Okay. We're going to do an activity to illustrate these points, all right?
누가 되기 때문에 의견 중이세요.
We're going to have 3 inputs to the neuron.
자네 먼저 저번 주에 가서나에 다친 숨을 주고 그랬어.
You guys have input number 1, come on up.
기란소에 다친 얘기들다. 미숨 강한 미숨.
Well, you're number 1, make a line here.
사람들이 반 리치.
Your input number 2.
기란소에 이바들다. 다친
Make a line right here.
You guys, you make an input here.
숨 소리.
Number 1.
Stand on this line, right on this line.
Good.
Let's just have 2 inputs.
No.
Okay, 3 inputs. Oh, okay, 3 inputs.
Okay, number 3.
Can we have them over here?
Cell body.
Okay. So you go right here. Input number 3. No, here. Okay, get a cell body. You guys, this row. You're the cell body. Make a circle right here. Circle.
You're the axon donor.
그럼 물총 줘야 된다.
We'll explain what you do in a minute.
Okay,
You guys come here. You
so-
stand down this way.
We need you here.
기란소 this bonding.
Maybe a little bit of a membrane here.
Dendrites.
Dendrites, right. Okay, you guys come here.
You're here. You're a dendrite.
기란소 달린 사진이다.
Dendrite. 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 We need one more.
So you're a, what are you?
진도를
Axon number 1.
Axon number 1,
Axon number 2.
axon number 2.
Axon number 3.
Okay?
So they get down like this.
Or,
기란소 막 디스토션인데 그랬어.
okay, you're going to fire an action potential.
기란소에 레직 독스 펜 거다.
Okay, so now, me too, I guess. Up. Brrr. Okay? And,
Do one at a time.
oh, okay, one at a time. Down again, down again.
냠도 많이 받지. 림벨 발음 쓰네.
Okay.
1,
찍질라 찍.
then 2, 3, and 4. Then you tap him.
Yeah.
Tap.
콜라도 쓰고 그랬어.
You tap where the cell body here. Tap him.
조금 늘들어 쓰고 그랬어.
Now, this wasn't enough to get it wasn't high enough. We need some more input to get the action potential generated.
레직 독스 전야가 당연히 막 있다 다쳤는데 좀 봤어.
I know. So the next one, when we add 2, he 2 people touch him. Exactly. I agree completely. So we have to have all of us jump, and when we have all of us jump, he will be strong enough to make him jump. We all get down. Okay, we all get down. Okay, choreograph. This one by itself.
도서시는데 지금 냠득지 웃었다.
No, just this group first. Just us first. Go. 1. Go. 2. This is an action potential arriving. Okay? And I hit him. But now nothing happens.
We only need 3 of these because there are 3 dendrites. Okay. Just a second. Okay, good. Dendrite. Alright. If the person behind you taps you, you tap him.
저는 수수 경험이 주시듯 한번 등기면 들으시고 그랬어.
So you want us?
Only if all 3 tap you do you jump.
저는 미 숨기 결혼을 주듯 한번 시내에 결혼을 해야 한다. 미 숨 마찬가지 결혼을 하는 말이다.
Okay, also, they have to understand that we're the action potentials.
결혼에서 정말 레지 독서다.
We're traveling.
그래서 주시적으로 로고로.
That's why it jump. It travels. The action.
찍기질래 찍도서 야라는 로고로 왔어.
And only when we have 3 action potentials arriving will it be strong enough to make a new one go.
그래서 순간 염도 저런데 정말 시내에 레지 독서 중요하다 당연히 마찬가지.
Okay, you jump if only all three. Okay?
저는 기랑라야 디크스나 미숨 기랑라 냠도 수스나 데네 시네 기랑라야다.
Let's try this row. You action go. 1, 2, 3, 4, 5, tap in, tap in, tap in, tap in. Well done. Good.
그다음에 야나무 말아.
So now we go down again.
Okay. Let's try 1 and 2.
니
Not 3.
숨 말다 니.
No.
No, not enough.
1 and 3.
자, 순간 냠도.
Chumama. Up, 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 up.
Okay, great. One more time.
So the hillock is supposed to tap the next guy.
Yeah, that's it.
He just jumps. Okay.
Then
그다음
the so-
그럼 물준데 야람데 수가 기랑선 붙은데 야람 보다.
Okay, you understand? This is the input. This is the dendrites and soli. These the axon hillock, and this is the axon.
저는 내일 가서 그 하면서 랩 사이드다 랩 영 사이드다 네 다지 랩직 독체 들어. 저는 내일 가서 가고 리체 드네 내일 가서 물촌체 드네 다게 쓰는데다 다게 쓰는데로.
Okay, 1, 2, 3.
순간 냠도.
So the process, we're now going back to what I was supposed to be talking about, which was chemical transmission.
저는 제줄 충동해야 되네. 그저 다른 데가 있마다 이제 쇼미스.
So there are three things that you have to have
You have to have for neurotransmission.
for chemical
저는 제줄기 충동 그 되라. 네, 이거부터 참고해야겠다. 네, 그래서 무슨 뉴레스.
release of neurotransmitter,
아까 가서 언더 춤을 그랬어.
binding to receptor,
저는 닐림바가 잘해야 되니 맞지? 닐림바가.
and ending the interaction.
다음에 충동되는 죽죽 그랬어.
You understand?
저는 벌써
Release,
뭐
binding, termination.
언더 춤을 언더 춤을 이제 고 말에 닐림바가 잘. 근데 그 기타 제조식으로 제조를 되는 배워서 죽죽.
Okay, so one thing that we haven't talked about is termination.
저도 다 죽져야 되면서 다다 볼때 계제시야 그랬어.
When you release neurotransmitter
언더 춤을 리드 참바스.
and it travels across the synaptic cleft
아니 뒤쪽 발동도 거기로 왔어.
and it binds to the receptor,
닐림바가 잘.
there's a lot of neurotransmitter around here.
그래서 그 예율로 언더 춤을 시험 공부해 왔어.
You have to get rid of it.
저도 다 언더 춤을 이제 고 배워서 그러 왔어.
Why do you have to get rid of the
같이
neurotransmitter?
배워서 그랬어. 언더 춤을 이제 잘 와. 이 제가 같이 배워서 그랬어. 맞다. 나름 속에서 so 나름 to inhibit
To end the signal
저도 다친 데고 주대야라
or and to allow another signal to be sent.
다 영나 다친 셴바직 줘야라 네 다다디 원자 좀 받기 배워서 그랬어
So there are two ways to get rid of neurotransmitter.
저도 원자 좀 받기 배워서 탈론이 그랬어
One is to cut it
지금 가서 그 원자 좀 먹고랑 동부 땅에 그랬어
so it no longer functions.
저는 그 질레나고 잘 그랬어
And the other is to gather it and put it back into the neuron.
이번에 가서 그 원자 좀 받기 래 네 원자 차고 나고 다시 그랬어
So there is a protein that does this, the transport.
들리는 거 원자 좀 렌션에 어린이주라 워킹 데이터에 기재지 그랬어
It's called a transporter.
띠밀라 갈수 것은 기름 봐서 그랬어.
If this is the presynaptic terminal,
기자들이 뭐 두숨 먼저 중내된 바이나
and I am a transporter,
아니 나 기름 바이나
I'm standing here with one side in the synaptic cleft,
그다음에 나 두숨 방송라 대체네 니 들라 갈수나 두숨 전로기 중내되로
and I try to grab a neurotransmitter
그다음에 갈수나 완전 집어질래
and put it inside.
그다음에 완전 저번
Grab a
전로기 중내되로 나로 간셔로
What does this remind you of?
디 트란스라 정말 갈수 가래다 나오도 미치다 펌
It's a kind of pump, okay?
미치다 나와요 그래서 미치는
You can think of the transporter as a vacuum cleaner.
vacuum cleaner.
You know what a vacuum
예.
cleaner?
어디는 기름과 되게 되면서 짱마성이 주제지다. 짱마성이 주제지다. 어디는 이제.
Okay? So it takes the neurotransmitters, puts it in. So it's like a vacuum.
완전 tumba디 이들래. 네. 완전 저거 나오고 딱. 완전 tumba디 이들래. 완전 저서 나오고 딱 거였어. 짱마성 거였어.
Okay, so here we have a picture. Just to review again, this happens to be a dopamine neuron.
디카스나 도파민기 완전 저서 됐어.
What are these called?
이게 되면 민가스를 어디 gambo vesco.
And how many dopamine molecules are inside?
gambo 나올라에 도파민 기제가 되래. gambo 나올라 도파민 가져래. 1,000 to 2,000.
Right. And then you have a signal arrive, and then you have this fuse.
그래서 다친 딜렉체 내고 기모 같은 로라 나의 넘어지.
It travels across the synaptic cleft, it binds to the dopamine receptor.
그래서 완전 정말 이제 뒤쪽 방금 견에 이 넬레바들 렉체 넬레바가 잘 그래와.
Will dopamine receptor bind glutamate?
근데 도파민데 dopamine receptor. Okay. 근데 glutamate dopamine की 넬레바가 잘 그래와. Glutamate dopamine की 넬레바가 잘 그래와.
No. Each receptor is specific for a neurotransmitter.
저한테 그 넬레바 수수수가 가서 왔다 잘 보세요. 다음에 완전 정말 먹어봐야 돼. 저한테 남 냄새 진짜 보여.
So of course if you have glutamate being released,
딱 이제 글루타민 들어와이나.
you have to be a glutamate receptor.
저한테 글루타민 넬레바들 잘해야마도.
If you are dopamine, if dopamine is released, you have to be a dopamine receptor.
저한테 도파민이 넬레바치가 잘해야마도.
Okay, so we have dopamine vesicles that fuse, they release the dopamine, they bind to the receptor, and this is the reuptake transporter. Okay, so I told you that it is important to remove the excess amounts of dopamine and you either cut it in half, there are enzymes over here that can cleave it if they go down too far, or or the dopamine reuptake transporter turns on and it brings the dopamine into the presynaptic terminal. So why does it do that? Two reasons. One is to get rid of it from here. And the second is that it gets packed back into vesicles. Okay, so now certain drugs that are bad for you. Have you heard of cocaine?
그래서 나 코카인에서 이제 저 타만하고라 타만하고 그래.
Cocaine.
코카인에서 타만하고 그래.
You've heard of it, a drug of abuse.
그래서 나 이제 타만하고
You can develop an addiction.
들라 가스나 타만하고 들라 이제 가서 우리 짤랑 멤버 쇼르다.
How does cocaine work?
네, 코카인 길레가 변리시 버렸어 왜냐?
Is shown here on the right side. What is it binding to?
Dopamine receptors.
No. Where are the dopamine receptors? Here. What is this?
Transporter.
The transporter. So cocaine binds to the transporter and blocks it from working. Looking at this picture, tell me what the blocking of the transporter does.
So the excess dopamine cannot enter back.
Exactly. There's too much dopamine then. Okay, now we're turning slightly away. We're still looking at a axonal tip. This one has vesicles with neurotransmitter, and so does this one over here. But in this case, the receptor, let's say glutamate receptor, is an ion channel. So when the glutamate binds to the glutamate receptor, the receptor channel opens, ions, not the neurotransmitter, but ions like sodium or potassium or calcium flow in. So is this okay, this is one type of one way that we can activate a postsynaptic terminal. Other types of receptors are shown here. It is binding to these proteins next to it. There's no ion channel here, but these proteins have activity of their own. And they then have to move across the membrane to activate, for example, this channel. Or this protein, or this channel. So a neurotransmitter is released and binds to this receptor. Then this protein is activated and travels across here. Does something to this channel to open it, or this protein to make another protein or another message. So here's my question: which one is fast and which one is slow? A is very fast. B is longer. So why did we come up with B to do something? The reason is that this ion channel has one function.
이 점수 가르스나 다 기들 결론디 기리들라 그 칠레치 레스 칠레치 말레스.
Sodium only, or potassium and calcium go through. There are ion channels. So the one function is ions go through.
저는 그 칠레치 가르스나 그 기들 결론디 기는 그 기들 교토 그레스 기들 그레스.
This receptor, which is slower, but it's more, has many other things it can do.
다 넬레바 디기리들라 그 대우 요레스 이바이나 그 칠레 샤바 망고들이 치토 그레스.
It can activate other proteins that are here on the membrane.
다 기모가라 이 칠레 샤바 디라인에 셀론 치토 그레스.
And those other proteins are required or necessary, for example, to strengthen. Remember that we were talking about strengthening synaptic contacts?
저런다 제제디기 가르스나 그 뒤층기 뒤줄기 바로 뒤줄기 템무소 그래라 봐. 저다 그 템무소도 그레스.
So that's one of the functions of those other types, those slow receptors.
저런다 넬렘바 대부디기디 네 칠레 샤바지 디샤 그레스.
Here I'm just showing you again. In this particular example, we have a neurotransmitter arriving. Binds to a sodium channel. It goes now, it opens. And in this case, both when it opens, ions flow through. In this second, the slow transmission, here we have the transmitter. It has its receptor. It's bound to this protein over here. When it's bound, it activates this protein. It travels across the membrane and it activates this channel. Okay, this is just summarizing what we've talked about. Neurons communicate with one another through synaptic transmission. Neurotransmitter is released. The neurotransmitter binds to specific receptors. And these receptors can either be excitatory or inhibitory. We're going to stop now. I was going to show you a movie, but I'm not going to show it because it's now tea time. When you watch movies here, do you watch movies sometimes? Do you bring popcorn? Okay, so bring popcorn back after the tea. And we'll watch a movie. Okay, thank you.